First off, I would like to thank the organizers for giving me a chance to present here at the NALMS 2020 Virtual Symposium. My name is Gary Anderson, and I'm the Chief Technical Officer of Watershed, a DNA testing company that provides smarter lake management through innovative methods in microbiome technology. I also have a joint appointment as a professor of environmental science at UC Berkeley and a senior scientist at Lawrence Berkeley National Laboratory. I thought I would share a picture of myself here up front so that you would have an idea of what I look like, rather than having a little corner video of me throughout the presentation. Today I will be talking about a scalable approach to identifying all fecal sources of contamination in watersheds. And since this is a lake conference, I will be focusing on lake contamination. With human encroachment into pristine locations, more common extreme weather events, and increased agricultural stresses on land, we are seeing more and more fecal contamination events in our waterways. A recent study found that over half the beaches in the U.S. had significant levels of contamination. As we see from these headlines, this water quality degradation is happening in our freshwater lakes as well, such as in Wisconsin, where a number of extreme storms since 2018 have caused massive flooding events and fecal contamination problems. The degraded water quality in these lakes has a number of negative consequences, including increased disease exposure risk, loss of recreational opportunities, decreased home values when you can't access the lake, and potential litigation when someone is blamed for the problem. All current water tests are unreliable and at best provide only preliminary data of potential contamination. Most tests are not able to identify the specific source where the pollution is coming from. They are inefficient because there is a reluctance to take any action due to a general consensus that the results are often of questionable quality, and they are very inaccurate with many false positives and false negatives. One of the main drawbacks of the current methods, from the simple culture-based test to the more sophisticated test using the latest DNA technology, is that they all rely on the results of identifying a single indicator microbe that serves as a proxy for contamination or specific fecal source. If that indicator organism is present in a fecal source at lower than typical levels, then it may be a false negative or not detected. Or if a related environmental organism that grows outside the fecal source is present, it may be a false positive and wrongly indicate that contamination is present. The reason for this is that the microbial community in lake water is very complex, with tens of thousands of different types of organisms. Let me illustrate this in the following crude diagram. Here we see a representation of a cross-section of a typical lake. I have included a number of different styles of green shapes to indicate the diversity of microbes that are always present in a healthy lake. In reality, there could be over 10,000 different species of microbes that are present, each in a very, very small quantity in the lake water. These are not harmful, they are not pathogens, and in fact, the lake could not function without them. In addition to these healthy lake microbes, a number of different algal species may be present, as indicated by the blue symbols here. The time of year or weather patterns may favor the growth of some over other species. In addition, some microbes may only be present under certain chemical or physical conditions, as indicated by these purple symbols. If there is contamination from a specific source, say horse fecal pollution, then there will be hundreds to over 1,000 specific microbes to that particular source, as represented here with the horse figures. Similarly, if a human source, such as septic waste, is present, there will be hundreds to thousands of microbes specific to this source, with some overlapping with other human sources, as seen by these red symbols. Finally, if any fecal source is present, then a very small fraction of that source may contain pathogens that are harmful to humans, as indicated by these pizza slice symbols. Although it is important to remember that this is a small fraction of an already small overall amount of total microbes in the water. This very large and diverse collection of microbes in the lake water constitutes what we call its water microbiome. And that is our focus at Veriset. We can detect all sources of contamination in one test using microbiome DNA. Our phylochip DNA microarray has the unique ability to rapidly identify all microbes in a single small water sample. By identifying hundreds to even thousands of microbes that are specific to a single source, we are the only method that is able to use statistics to measure confidence. How the phylochip works is that it targets a region of DNA that is present in every type of bacteria. This region can be thought of as similar to a grocery store barcode. Each bacterial species has a unique DNA sequence that is used to differentiate it from other closely related bacteria. That region is represented here in the top figure, where the red represents a DNA sequence that is shared among all bacteria, and green is a sequence that is unique to that particular species. The phylochip identifies all the variable sequences so that it can identify up to 60,000 different types of bacteria in one sample. We have identified all the bacteria that can be found in each fecal source type, including the species that are unique to that source. There is a series of machine learning algorithms that combine this information with a complete microbiome analysis of a particular water sample to computationally identify the probability that any specific fecal source is present. So in other words, we have extreme confidence in our source detection. We have analyzed the signatures from many different fecal types. Here you see some pictures of our interns collecting samples from a variety of species. We've also analyzed samples from different types of human sources, including sewage, septic waste, and fresh human waste as examples. Our methods can actually distinguish between these different types of human pollution. We have also collected and analyzed sources from a variety of domestic and farm animals, wild animals, birds, etc. We're now up to around 40 different types of sources that we can use as a baseline to detect their presence. We have already used this technology to solve a number of real-world problems, and some have even been significant enough to be mentioned in the local press. For example, in Malibu, we were able to identify the source of contamination that was impacting the local beaches. We were able to identify that wild animals and not humans were responsible for watershed contamination in Kauai. In all these cases, we were able to bring clarity when people had been arguing with each other for a long time about who was responsible. The Russian River is considered an impaired recreational river of Northern California by the EPA. Here we see a map where the main blue line is the Russian River that flows into the Pacific north of San Francisco. What is circled in red is the location where we found the primary pollution on the river caused by failed septic systems from overused vacation homes. The green circle shows the location of dairies where cattle signatures were found on this tributary. The purple arrow points to where the river meets the sea, and huge numbers of seagulls here were the reason we detected this signature. What is really interesting is all along the upper half of the river, the black dots were locations where we were able to show that there was never any contamination, even though it had some of the highest fecal coliform counts. What happens when you have a crowd of people in or next to a river for 2 days to listen to a jazz festival? We found out that you may not want to know the answer to that. As you see from the graph on the upper left, a morning water sample was taken 5 days in a row, including before and after the 2-day concert. The red bar on Sunday indicates significant human fecal contamination after the first day of the concert was over. The even higher red bar on Monday represents one of the highest levels of contamination that we have measured. The standard fecal indicator test did not show any actionable levels of bacteria until Monday, when it was just over the threshold. We often see a disconnect between our fecal assessments, which are based on statistical validation using more than hundreds of simultaneous independent confirmatory observations, and the commonly used standard fecal indicator test. In the graph shown here, we compared the values from a standard E. coli fecal indicator test, shown here on the x-axis, with the measured level of E. coli that was obtained from the phylochip for each of about 60 water samples. The samples where we had statistical validation of fecal contamination by human waste are indicated by red squares, or by cows are indicated by green triangles. The samples where the phylochip identified a healthy water microbiome are shown as black circles. Overall, as the E. coli signal increases by the standard test, it also increases by the phylochip, showing agreement of the E. coli values in both types of test. Anything to the right of this light blue dashed line represents a water sample where the E. coli test would say that the water is contaminated. We see phylochip results that show contamination both above and below this threshold, as well as a lot of black circles above the thresholds. In other words, there's a lot of false positives and false negatives with a standard test. This is why we do not trust the results from the single fecal indicator tests. Although most of our work to date has focused on large watershed scale projects, Veriset sees the value of using its technology to aid in lake water quality management. The challenge is having a user that has no formal water testing training taking a single lake water sample for analysis. The methods required to collect the sample are actually reasonably straightforward, and shown here the first step is to collect the 100 milliliter water sample and then to concentrate the microbes onto a filter. Again, showing the first steps are to collect the water sample and then to concentrate by passing it through a filter. The reason for the filter is that it is easier to mail a small filter than a larger water sample. The next step is to send the sample by overnight mail to Veriset, or if you happen to be doing several samples at different times, you can place them in any freezer until you are ready to ship. Veriset provides a kit containing a collection syringe and filter to individuals that want to take a sample. So what do you get back when you send in a water sample? Here I am showing a mock sample report that has all of the pertinent information about the water sample and whether or not it passed quality control. In this example, the water sample had contamination from septic waste, as well as a lower concentration potential signal from geese. We are still figuring out how to display this for someone that wants to analyze just a single water sample. But one thing we are thinking about is color coding. So red for a statistically significant level of contamination, yellow for potential contamination, something that may pose a problem in the future, but does not rise to statistical confirmation at this point, and green, meaning that you have a healthy water microbiome. We could also include bar charts to signify the relative amount of contamination. Similar to genealogy-based DNA testing companies like 23andMe, we actually possess a huge amount of information that could be mined from each sample. For instance, you could figure out what types of algal species are present or how the water microbiome changed after a chemical treatment. Freshwater harmful algal blooms are often caused by cyanobacteria, also known as blue-green algae. Some, but not all species of these algae produce toxins, such as microcystin, that are harmful to humans and animals, but it is not clear what triggers them to suddenly produce the toxin. Our phylochip can detect 150 different species of blue-green algae, and it may be possible to use this information to help inform when to treat. The figure here shows the different concentration of the individual species that may be present in a lake water sample. This information could provide one more piece to the puzzle about if your lake is going to get an algal bloom or a harmful algal bloom. An example could be to see if a specific algal species change between the samples taken 1 month and the next month. Although this represents an exciting opportunity, we do not have any specific examples where we've done this. At this point, it is just an additional piece of information that a lake manager may track to see how it actually correlates with a lake's HAB status. In conclusion, I hope I could convince you that the lake water microbiome presents a number of unique opportunities for lake management. All of the current single microbe indicator tests provide questionable information. In contrast, using the entire collection of lake microbes provides a highly confident information on sources of contamination. And finally, identification of presence and relative concentrations of 150 blue-green algae species could potentially be used to decide when to treat to prevent harmful algal blooms. With that, I'd like to thank you for the opportunity to speak, and if you have any questions later, please go to the website below. Thank you.